Dr. Vavali to please introduce our today's speaker. Please, sir.
Very good morning to all of you. It's my pleasure to introduce our today's speaker, Dr. Pillaiyar. Sir's research interest is in synthetic organic medicinal chemistry, total synthesis of natural bioactive molecules, methodology and development for synthesizing novel organic molecules with potential biological applications, and drug discovery and development. Sir completed their education as follows, doctoral studies in pharmaceutical chemistry, in design synthesis and biological evaluation of novel thiourea analogs as melanogenesis inhibitors in South Korea. Sir completed their Master of Science in Chemistry in Tamil Nadu, India. Sir completed their Bachelor of Science in Chemistry in the University of Madras, Tamil Nadu. Sir is currently independent junior research group leader at Tübingen, Germany. Sir is also visiting scientist. Sir was also visiting scientist in University of Cambridge, United Kingdom. Senior scientist in University of Bonn, Germany, and received a prestigious Alexander von Humboldt-Bayer postdoctoral fellowship in 2013 to 15 in University of Bonn, Germany. Sir also had the industrial research experience in various industries. Sir's main key research area involved infectious diseases, cancer, inflammation and neurodegenerative diseases, muscular dystrophy, heart failure, and skin diseases. Sir has various awards and recognition on his credits, including recipient of the International Conference Grant from the ACS Fall 2020 National Meeting and Exposition, Chemistry from Bench to Market, Short-Term Scientific Mission Grant, DAD Grant for the traveling to present their research in conferences, Chemistry Frontiers and Challenges. In the conference, Sir was invited as a guest speaker. Sir also received the Alexander Humboldt Fellowship and many more in his credit. Sir is a member of Pharmaceutical Society of Korea, Chemistry Society of Korea, the Japanese Peptide Society, member of the German Purine Club since 2014. Sir also had the experience in teaching and mentoring from 2008 to current years. Sir had the many publications on his credits in the reputed scientific journals, including Angewandte Chemie, Journal of Medicinal Chemistry, Organic Letters, Journal of Organic Chemistry, ACS Omega, European Journal of Medicinal Chemistry, Bioorganic and Medicinal Chemistry, and many more reputed journals of almost of all the publications. Sir also the peer reviewed for the various journals, mainly the Natural Chemistry, Biology, Journal of Medicinal Chemistry, Journal of Organic Chemistry, and many more. So we are really honored to have this great personality today in our International Faculty Development Program. And I sure that you will learn many more things in this talk. Now I request Dr. Pillaiyar sir to start with their session.
Okay, thank you very much for your very broad introduction. So I'm very happy to be part of this conference, and I'm basically a medicinal chemist, so working on various fields, and yeah, mainly on the coronaviruses and especially the SARS coronavirus 2. So probably my presentations is focusing on especially the SARS coronavirus 2, how the natural products are fighting against coronavirus disease. So thank you very much. Okay, then let me start. Okay, the content of my introduction, or I would like to talk about the SARS coronavirus, human coronaviruses, genome, disease, life cycle, mutants, especially the SARS coronavirus 2. Then I would like to focus only focus on some targets and how the natural products are intervening the viral infections. More exclusively, I'm going to talk about the viral entry inhibitors, that is modulation of spike and other proteins. And there are cysteine protease, which are very important in the replication of the virus. And finally, the conclusions. So basically, the coronaviruses are classified according to the International Committee, according to the International Committee for Taxonomy of Viruses. So if you take our human coronaviruses, they are classified under the subfamily of coronavirinae, which is under the main family of coronaviridae, and that in turns under the order of nidovirals. So these coronaviruses can be further classified into 4 different genus that are alpha, beta, gamma, and delta. As you can see here, the viruses that are highlighted in red colors, they are highly dangerous. They are responsible for 3 global pandemic, especially the SARS coronavirus 2 is still important, even though it has started 3 years ago. So basically, the alpha and beta coronaviruses infect only humans, and gamma and delta coronaviruses infect birds and animals. The coronaviruses are believed to be transformed from the bat through different intermediate hosts. For example, the SARS coronavirus 1, which was emerged in 2002, end of the 2002, was believed to be transferred from the host intermediate civet. And for the MERS coronavirus 1, which was believed to be transformed from the camel, and for the SARS coronavirus 2, it was a kind of hypothesis that it might have spread it from the pangolin from the bat. So far, we have identified 7 human coronaviruses. The first 4 human coronaviruses are responsible for 1/3 of the common cold in humans. The other 3 beta coronaviruses are highly dangerous. For example, the SARS coronavirus 1, which was emerged in 2002 in China, MERS coronavirus, which was emerged in Middle East region, and the current ongoing SARS coronavirus 2 was emerged in 2019 in the end of December, and it is still ongoing, though huge efforts have been made to contain these infections. And of course, this is the reason for the ongoing global pandemic. The SARS coronaviruses mainly affect the upper respiratory part through the nose and maybe by the liquid tablet. So it spread to other tissues like lung, epithelial cells, and in the severe cases it goes to kidney, heart, brain, and this is the kind of overreacting or insufficient reaction of the immune system. Then that is called cytokine storm. So that cytokine storm means it produces the uncontrolled pro-inflammatory cytokines, which affects the normal cells, and that leads to the organ damage. And finally, in severe cases, that can also be reason for the death. So the other problem with SARS coronavirus 2, that is the reason for the COVID-19. When you have the long COVID-19, then that you will have always the mild headache, even after the clearance of the virus. Sometimes you get the fatigue and loss of distorted smells, and sometimes you always feel like muscle that you are very weak, and you feel like always low fever. So this is not something that when you take the tablet or when you clearance the virus that you are completely clear. So you need some times. So this is something unprecedented effect that have been observed for other coronaviruses. So this is really very serious. And then, sorry, yeah, okay, great. Okay, so it's very important to understand in order to develop the vaccine or some therapeutic molecules. In order to develop therapeutic molecules, if we really understand what is the life cycle of SARS coronavirus 2, how it is really involved in the infectious process. Here I classified into 9 different stages, but it can also be more. But I hope this could give you more important insight in the virus replication cycle. Take it. So The first step is binding of SARS coronavirus 2. The SARS coronavirus 2 recognize the receptor in our host system, that is ACE2 receptor. Once, once it is attaches into the cell receptors, it will be internalized. That means the decoding, uncoating of the viral RNA. The viral RNA is then ready for the translation process. And that produces two viral polyproteins that are called PP1A and the PP1AP, and which is very important in the replication step. And these polyproteins can be cleaved or proteolyzed by two viral cysteine proteins that are called papain-like proteases or main proteases to produce the non-structural viral proteins. That includes also the RNA dependent and RNA polymerases, which is catalyzing the catalyzing to produce the genomic RNA. So basically in the RNA replication step, the negative sense RNA genome can be used as a template to produce a positive sense genomic RNA. And you have the mRNA, RNA of structural proteins. This can also be translated to the structural protein. They all pack together and then assemble and then exocytosized from the infected cells to another infected from the infected cells to normal cells, which can continue to infect the other normal cells. So by intervening any step on this process, one can create a very effective therapeutic options in order to contain the viral replications. I don't know why it's not. Okay, so this is not only the other problem, because we have also a lot of variants. The variants means it's nothing but is the mutation of the mutations in the spike proteins, because which is important to recognize the host receptor. And when you have the mutations, and then it has the different properties. For example, we have different kind of variations from alpha to Omicron. And based on the mutation, it has a different capacity to infect the people. For example, the alpha mutations, which was found in United Kingdom, which was found to be even 50 to 70% more infectious than the wild type that was originally emerged in China. And we have also the beta coronaviruses, and gamma was not that dangerous, but the delta, which was found in India, which was the highly dangerous and highly infectious, and that was the reason for probably the second wave or third wave of the India, which killed enormous people in India. And we have also Omicron, which is very infectious, but fortunately they are not very much virulent. So how they are, basically the coronavirus variants are classified according to the Panko nomenclatures, which is kind of the tool. As you can see the chart, they utilize kind of the genomic sequence of the coronaviruses, and based on the mutations, they can be classified as from alpha to beta, and this can be even more when you have the new mutations. So how can we cure the virus infections? How can we treat the viral infection? So basically we have two options. One is preventive, that can be used by the vaccination. So it can be mainly targeted the spike protein, and we have different vaccines available already. The other option is the treatment. It is only after the viral infection. So once you recognize that you're infected with the virus, then you have the different options. For example, antibodies, which is also targeting mainly the spike protein, different region of the spike proteins, and the small molecules that could be natural producers and other molecules. They are targeting the other viral proteins and also the host and also the host proteins that supports the virus replications. So we have more than 20 vaccines are available right now, and more than 70% of the world population has already received at least one dose of the COVID vaccine, or at least more than 13 billion doses have been administered globally. And every day is more than 100,000 doses are administered in order to contain the SARS coronavirus infections. And of course, we have seen the huge development, though the pandemic is ongoing, but almost all of the country, they lifted the many restrictions. So we are still free because we have the very effective vaccines. So by using the monoclonal antibodies, we have different monoclonal antibodies in order to contain the SARS coronavirus infections, and that can be used for the patients with mild to moderate disease or even at the high risk of severe progression. And we have seen really the reduced risk of hospitalization or death, and maybe the faster resolution of the symptoms. We have also the human monoclonal antibodies that are approved under the emergency authorizations. They target different regions of the spike proteins. And we have some example here, the casivimab, pomlanivimab, and then soravimab. They are very important monoclonal antibodies, but they are very difficult to inject in the hospital condition, because you really need to maintain the monoclonal antibodies concentrations, which is sometimes very difficult to treat. So what is the other option? What is the other option is treating the viral infections. When the coronavirus emerged, the scientists have utilized several approaches. One of the approaches is repurposing the old drugs. Old drug means that are approved or maybe stopped due to the clinical safety reasons. For example, lopinavir and the ritonavir, which are approved for the human hepatitis virus and the human immunodeficiency viruses. So what is the advantage? The advantage is one can easily develop the drug, and maybe they reduce the risk of failure due to the safety, PK, or reduced cost. So you don't have to really start from the scratch, because you have already available the approved drugs or the drugs with completely characterized. So one can easily dissolve the problem in the initial stage. The other option is completely start from the new scratch. You have to identify the target, basically the viral target, the enzymes that, for example, the polymerases or proteases that are very important in the virus replications. The advantages is when you target the viral target, it should be very specific, so that the side effect also will be very less. But the disadvantage is you may have the resistance to the development, resistant to the target, because the virus gets very easy mutations and it's very rapid. So if you target the viral proteins, then you have the chance to mutate. Then that means you have to really create again from the scratch. So this is what we are having the problem with the spike proteins or with vaccines. So when you have the different mutations in the spike proteins, so the vaccine that is actually targeting the spike protein can no longer be used, and then you have to make another vaccine, which is very expensive. Another option is targeting the host proteins. So usually there will be less chance of resistance, but the disadvantage is the side effects, because when you're targeting the host proteins, that means you're also interfering the normal process of our host proteins, so you get the side effect. So this is also the problem when you're targeting the host proteins. So far we have 3 approved drugs for the SARS coronavirus 2, which is causing the COVID-19. So this is the simple version of the virus replication cycle. The one drug that is called Paxlovid, the chemical name is nirmatrelvir, which is targeting the main protease. As I mentioned in the virus replication cycle, it is one of the very important enzymes which catalyze the polyproteins, and that produce the non-structured proteins, which are important for the further replications and for the production of the transcription complex. So when you block the key enzyme that is called the main proteases, you will you will stop the replication so that the virus can be killed. There are also other two drugs that are called Vicluri, that the remdesivir is one of the very famous drugs that you have heard about it, and the molnupiravir, which was recently approved. The both drugs are targeting the RNA dependent and RNA polymerase, which is producing the viral genomic RNA. But when compared to nirmatrelvir, they are not as effective as nirmatrelvir. So these are three drugs that are approved right now, and then many more drugs are under the clinical trials. Okay, now I come to the topic, the real topic, that are natural products against the SARS coronavirus 2. So those were the backgrounds about the SARS coronavirus 2, so that it will be easy to follow for the upcoming slides. So natural products are, of course, is very important sources, and they have a very low molecular weight and the minimum toxicity. So based on the information that we had for the previous coronaviruses infection, for example, SARS coronavirus 1 and MERS, so we have already the natural products that were implemented against the other SARS coronaviruses that can also be used. So that can also be used for the novel virus infections. The other good thing is the natural products, which is basically in a complicated structure and maybe very rigid, that they can also inhibit the other coronaviruses in different stages of the virus replications. And indeed, when the SARS coronavirus 2 emerged in China, many herbal medicines have been utilized. Some of them are camellia, allium, and ginger, the compound kind of the gingerol, and scuturilla. I can say so many examples. They have been utilized in order to improve the immune response to contain the SARS coronavirus 2. So now I would like to focus on the specific targets, basically the viral entry and the other cysteine proteases. So if you look closely into the viral entry, as I mentioned, that the SARS coronavirus 2 utilizes as a spike protein, which is basically divided into two parts, S1 and S2. The S1 part is mainly for the receptor binding, and the S2 part for the fusions. And you have the receptor domain, which the receptor domain is responsible for binding into the ACE receptor, which is the receptor for the SARS coronavirus 2 entry. So when it recognizes the ACE receptor, then you get the other transmembrane proteins, that is transmembrane proteins ACS2, which together with furin here, this is also kind of sub cofactor which can help the transmembrane proteins to help in cleaving the spike proteins. So this is how it is cleaving in order to facilitate the viral entry. There is also some other proteases that are called cathepsin B and L. They are not very important in terms of the viral, when compared to the transmembrane protein. So this is the reason for the fast entry of the viral virus into the host cell. So when you target the SARS coronavirus 2, there are I would say that the huge natural products have been investigated against the viral entry. So I would like to give you only some examples. For example, the capic acid, which is already well known for its antioxidant and anti-inflammatory and the other properties, which is isolated from the pavita and the indica and also many edible plants. So this capic acid was reported to inhibit the glycoprotein, that is the spike protein. So it's over here. This is the important step to enter. So it is reported to inhibit the glycoprotein attachment into the host cell. So there will be no more entry of the virus, and the virus replications will be stopped. This is one interesting example, and many other benzyl isoquinoline alkaloids such as tetrahandrine penquinolines and cephalosporin, they were already reported to exhibit the antiviral activity of other coronaviruses, other human coronaviruses, for example, human coronaviruses OC43. Mechanistically, they work by inhibiting the virus-induced cell death, by inducing the virus cell death via blocking the virus replications, basically by working on the SARS coronavirus, the spike glycoprotein and other end protein synthesis with host responses. And all three compounds are isolated from the Stephania tetrahedra. So more specifically, this cephalosporin compounds, which is also isolated from the same family species, which is known and it has been used as a traditional herbal medicines, and actually it is approved drug for several diseases with inflammatory and infectious disease. That includes also for the SARS coronavirus 2, and the compound was recently repurposed against the SARS coronavirus 2, and it was effectively inhibited SARS coronavirus 2 replications in the cells, which is very important, and also with an EC50 value of around 300 nanomolar, which is quite high, and showed a minimal toxicity. But so mechanistically, it works on the spike protein here, and then it binds to the spike protein and then stop the viral entry. So this cephalosporin is one of the very good example from the natural product sources. And then to continue, we have also several examples. For example, the tylophorine and 7-methoxycryptolopurine, they were also reported to inhibit the glycoproteins. And there is another compound, dihydrotranshinone, recognize the glycoprotein here of the SARS coronavirus 2 to block the viral entry. And we have other examples like luteolin and the TGC. So they are specifically recognized the YEST subunit, YEST subunit, and prevent the viral entry of the SARS coronavirus 1. More specifically, the luteolin also bind to the YEST protein to exert its antiviral capacity by interfering with the virus and cell attachment and consequent fusion. So it has the effect on the viral entry and the further the cell fusions, so that to be completely stop the viral replication. So it's very important luteolin, very important observations with the luteolin. And we have also example that targets not only the virus protein, that is glycoprotein, which is also targeting the human receptor, that is ACE2. For example, the compound imodine, which is one of the good example. So it works by blocking the interactions between the spike protein and the cellular receptor ACE2. That is, so it works on both humans and the virus target, and reducing the ACE2 receptor, ACE2 and transmembrane proteins through expressions in order to stop the viral entry. Very interestingly, this compound was also investigated in cells through the infected cells and then they showed a very good suppression of the infections in the in vitro cell assessment as well. The other compound, of course, it was aspirin, and the other compound was imodine, which is also targeting the human receptor interactions, and it also showed the antiviral activity in the cellular model. So these are two examples. So it was not only simply inhibiting the enzymes, but also the viral replications in cells, which is the cell is a complicated system. So if you could see the antiviral activity in cell, that means it's an indication that you could proceed with that molecules. So it's very important. So next, I would like to focus on the cysteine proteases that are main protease and the Papin-like proteases. I will tell you why it is important. So basically, the SARS coronavirus 2 is a single standard RNA viruses with the largest genome size of 30 kilobytes. So the viral genome encodes for the structural and the non-structural proteins. So we have two polyproteins from the from the viral entry, from the translation that are PP1A and polyprotein 1AP. So we have the 5' region and the 3' regions in the genome. And this is the region for this non-structured proteins. So when you have the non-structured proteins here, this can be cleaved after the translation. This polyprotein produces the two different polyproteins, for example, polyprotein 1A and polyprotein 1B, and these proteins with the different lengths can be proteolytically cleaved by two viral proteases to produce at least 14 different non-structured proteins. That includes also the RNA-dependent and RNA polymerases. So in this case, the main proteases plays major role because it has the responsibility to produce more than or at least 11 non-structured proteins out of 14 proteins, and the PL proteins proteases is responsible for producing about 3 non-structured proteins. The other end of the viral genome is responsible for translating to the structural and accessory proteins. We have different accessory proteins here. They are not only important for giving the structure, but it's also important to inhibit the immune response. The immune response in the host in order to facilitate the virus replication. So the structural proteins are also very important and the spike protein. Of course, this is what we had discussed before, how important it is, because it recognizes the cells by using the receptor in order to enter into the host cell, which is the first initial step. So when you look at again in the replication cycle, and you can see here, this is the place where the main proteases and the papain-like proteases plays important role that cleaves the polyproteins. And when you stop the main proteases or PL proteases, there will be no further cleavage of polyproteins PP1A and PP1B into non-structured proteins, and then it will stop the viral replication. So it's very important, and this is what the Paxlovid, which is the drug approved targeting the main proteases. So it's very important and very safe. Let me tell you more more in detail. So the main proteases, what is main protease? Main proteases is the cysteine proteases, which has dimer form. The monomer is inactive. So each monomer has 3 domains, 1, 2, 3, and they bind between the domain 2 and domain 3. So the catalytic site is located between the domain 1 and domain 2, which is very important because which catalyzes the viral polyproteins, so which has the catalytic diet system. So when you have the viral polyproteins, then it can be proteolyzed by using the different five different steps. But as I say mentioned here, so maybe you don't have to, look it into very detail. So, and we have also advantages of targeting the main proteases because it's highly conserved among many coronaviruses. So when you target one coronavirus, it can also be used for the other coronaviruses. And the main proteases play a very important role in the formation of the transcription complex that includes the RNA-dependent and RNA polymerases. So when you stop that, then you will really have the antiviral activity. And another important point is there is no human cellular proteases with a similar specificity. For example, the main proteases cleaves specifically when you have the peptide at the glutamine residues in the peptide. So if you have other residues in the peptide, probably it will not be cleaved by the main protease. So it is very specific and which is not available in our human normal cellular system with the same specificity. So when you target the main proteases, you will not interfere the normal process of our human cellular proteases. So it's very specific and selective targeting the main proteases. So mainly the compounds, mainly the compounds will be screened by using the fluorescence-based peptidic cleavage assays because we have the abyssal and the edens. One is the fluorescence activator and the one is quencher. So when it's intact together, when it's not cleaved, then it will be intact, and intact, but when in the presence of proteins, and then the proteins will main proteases, the peptides will be cleaved, and then this will go away, and these agents will give the fluorescence intensity, and this can be directly measured in order to measure the potency of the inhibitors. And for the antiviral activity, we have some requirements for choosing the in vitro cellular model. For example, when you choose the cell, then it must express the ACE2 receptor for the entry, because the SARS coronavirus utilizes the ACE2 receptor. And we have to also make sure that it has it expresses the proteases, for example, TMP-RRS2 and other purines. And you have to also choose the typical tissues that is affected by the SARS coronavirus too, because, for example, lung cells, which is the primary cell for the SARS coronavirus 2 to enter. And we have different cells that have been commonly used. That is the vero cells, and the Cowley cells is mainly arrived from the human lung cells that they express the ACE2 and TMP, RRS2 and other proteases. And we have also in the recent report that they engineered the different cell with the different genetic modifications to make sure that they express the ACE receptor and other proteases. So we have several options. And the antiviral activity can be tested only in the biosafety lab 3. That's the standard. You cannot use that in normal laboratory conditions. So it's highly dangerous. So indeed, several natural products have been utilized. For example, the upselins, bioropin, and so many natural products were already implemented against the SARS coronavirus 2, and they showed great effect on inhibiting the SARS coronavirus 2, and many other monoterpenoid compounds and ginseng compound, which is found in the eucalyptus plant oil. They were also potentially inhibit the main proteases inhibitor. So what happened actually during the outbreaks of the SARS coronavirus 2, there is a plant name called Isatis tankatera, and several other phenolic herbs were commonly used against the viral disease. I will tell you in upcoming slides how they are mechanistically working on this. And several other compounds from various sources, they were also reported. For example, some of them emodin, aspiratin, quercetin, and the dietins, they were all tested against the SARS coronavirus 2, and they show SARS coronavirus 2 main proteases and showed very effective against the SARS coronavirus 2 main proteases. Among these, the aluemodin, singerin, and hesperatins, they were from different sources. They inhibited the SARS coronavirus 2 main proteases. Then they also showed antiviral activity in cells in dose-dependent manner. Oh, sorry. Next, yeah, we have also some traditional Chinese medicinal plant, for example, Scutellaria, and from where the compound Scutellarin arrived, and it showed really very good broad spectrum antiviral activity against various coronaviruses, especially against SARS coronavirus. So it showed the EC50 value of about 0.74 microgram per milliliter, which was actually tested as a plant extract, and then later the compound also separated and then tested. That will be shown in the upcoming slides. And we have another interesting compound called bicalin that also strongly inhibited the SARS coronavirus 2 main proteases. And the other analogs of bicalin, for example, scutellarin and the dihydromyricetin, they have moderate to potent SARS coronavirus 2 main protease inhibitory activity in the micromolar concentration. So one of our colleagues or collaborators, they investigated several phenolic natural product that includes myricetin and dihydromyricetin, and they also synthesized basically based on these two compounds. So myricetin is a natural occurring flavonoids. Of course, this is observed in many edible plants, and it has wide range of bioactivities that includes antioxidant, anti-cancer, anti-diabetic. So based on this lead molecules, which is already very good in inhibiting the SARS coronavirus 2 main proteases with an IC50 value of around 600 nanomolar. So in order to improve the activity, one of our colleagues from China, they designed different semi-synthetic natural products. We know that the myricetin, and which is already showing the very good activity, which is also important to mention that they have antiviral activity in colloid cells, which is about in the micromolar, of course, but they are already very good with from 8 to 3 micromolar. In particular, the phosphate prodrug was found to be best one, which also showed good pharmacokinetic property, as well as the good antiviral activity in the cellular model. So they crystallized one of the derivatives, that is myricetin here, and they found very interesting aspect of the binding mode, that it bonds to covalently to the active site of the cysteine, which is something is unexpected with this compound, because we thought that there was no reactive group that can target the cysteine proteases. So not only the covalent bond, it established the various hydrogen bond interactions with the active sites, and they identified the pyrocolol, that is 1, 2, 3 here on the phenyl ring. So the pyrocolol mighty as a novel variant group that can be possibly engaged to make covalent bond with the cysteine proteases. So the mechanistically, so we have the myricetin here in the oxidation, in the natural oxidation conditions, it goes to the quinine form, and you have the catalytic active site here, histidine and cysteine 145. So when the histidine takes proton from the cysteine, and then you get the nucleophile, then the nucleophilic attacks the quinonide structure to make the permanent covalent bond with the myricetin. So which is very, very interesting in the when we observed for the first time that they have a very unique binding mode. So with the huge natural resource of the myricetin, and looks like very simple and low toxicity, and even with the unique covalent binding mode action targeting the protease, this can be proposed as a preclinical candidate for the further evaluations of its therapeutic potential in COVID-19 pandemic. And further, we continued recently, and then we were able to publish this year, in the beginning of this year, we investigated more than 100 natural products that have mainly active reactive molecules, something like Michael acceptor here, orodirunine, and polyphenolic compounds, something like this with rubinitin. So we found, for example, with the rubinitin, which is a bioactive natural flavonoid compound, which is of course flavonoid compounds, and we found when we tested against the main proteases, it showed really very good activity with the IC50 value of 0.95 micromolar, and then we crystallize this compound in order to ensure that really makes covalent bond with the pyrocolol unit. And you have seen here this cysteine 145, it really reacts with the phenolic pyrocolol moiety and makes permanent covalent bond. And we were also very curious to know about an already known compound, because the compound looks like very complicated, but still you have the reactive Michael acceptor group. Then we were very curious to test this compound, whether they are inhibiting the main proteases. As you can see here, the Michael acceptor is reacted to the cysteine residues in the active sites. Although it was weak, but it was very important insight to know that the compound is inhibiting the main proteases. So which is a kind of one covering titerpenoid net product isolated from the Rhabdosia rubescens, which has been widely used in the traditional Chinese medicine. And then to continue, we have also used EGCC. So I think you might know this is very important and one of the very famous compound which has been utilized in many different diseases. We also investigated this compound because we have the pyrocolor unit, and then we really see that it effectively inhibited the SARS coronavirus 2 main proteases, and by modifying the cysteine residues with the covalent formations. This compound also can also be effectively block the other signaling pathway, which is very important in the COVID disease, because this signaling pathway controls the production of the number of cytokines, which is reason for the cytokine storm in the severe cases of the SARS coronavirus 2 infections. So this compound also is very, and which is already very good in inhibiting the main protease. To continue here, we have also investigated the tricytine molecule. Basically, they are completely phenolic compound with, but the hydroxyl position in different, the hydroxyl substitution is different in different position of the molecules. That really makes sense. That will come up in the next slide. So we also investigated this compound, and this also inhibited the main proteases with the IC50 value of around 8 micromolar. So this compound is found in a variety of natural plants, and then that includes Leptiso, Perimedio, Chachas, Ecliptis, Globulis. So then what we did, we summarized all the active phenolic compounds that we investigated. For example, we choose the compound, for example, the cysteine, which is lacking the pyrocolor unit. But when we introduce the 5-hydroxy substitutions over here, then you really get the activity. But the counterpart cysteine is not active. So it's very important that you have the pyrocolor unit in the molecules. At the same time, we also introduced the other hydroxyl group in the ring A here, which is kind of beneficial for the activity, because you ultimately need the group that can facilitate the hydrogen bond interaction with the active sites. But when we remove this hydroxy position in the molecule, that leads to the tricytine, and then that means you lose the activity. So this hydroxyl group also is important for the SARS coronavirus 2 main protease inhibitions. Again, when I remove the hydroxyl group here, then you lose the activity. So it's kind of to make sure that it's really the pyrocolor unit is very important. But it doesn't matter where you have the pyrocolor unit, whether it's on the ring A or ring C. So we can see that it's this scutellin was found to be inhibit the main proteases with the IC50 value of 6.07, which is comparatively less to the robenatine, because you have the pyrocolor moiety in different ring. And also you have the pyrobicillin, which was also which is also shown in the previous slide, that they are inhibiting the main proteases with the moderate micromolar concentrations. We also found that it's very interesting, the compound with the myricytrine, we have the pyranose protection here. Though we have the pyrocolor unit here, but still the compound is inactive, and we propose that this could sterically hindrance disturb the binding of the whole molecule with the active site system protease. So this could be the reason why the compound is inactive, though we have pyrocolor unit in the molecule. So this is always also the same in the other. And as I mentioned, that the EG, CG, this is very important, because we have two pyrocolor units, so it doesn't matter that they can interact with the virus proteins. But when compared to this one, here there is a lack of carbonyl group, and which could also be the reason why it is active, because there is no steric clash between two carbonyl units. So it was very important observation. And finally, we concluded that the pyrocolal unit in the phenolic natural compounds is very important and identified as a novel covalent variant group for the inhibition of main proteases. we also found that other natural product compounds, for example, the resveratrol analyzed. In some studies, the resveratrol was reported to inhibit SARS coronavirus 2, but in our study, we did not see any activity. But its metabolite, that is called pisotenol, where you have the catechol structure, which is important, which is inhibiting the main protease when compared to its parent molecules. Resveratrol is well known for its various activity, because it has also the skin whitening activity in wine, especially in the grapes, that you have a lot of resveratrol. So that in the wine, the resveratrol helps to shine your skin. So that is also very interesting factor. But for the SARS coronavirus 2 main proteases, it is not inhibiting, but its metabolite, it is the hydroxylated compound. It's really inhibiting with low micromolar concentrations. And we also docked this compound to see how they interact with the active sites, main proteases. And we found that the catechol unit is really very important, because it engaged with hydrogen bond interaction with the threonine 26, which is lacking in the case of resveratrol compound. So it's very crucial for the inhibitory activity of the main proteases. And we have also other phenolic compounds, for example, the rosmarinic compound, which is naturally occurring. So this substance has been shown to have antiviral and antibacterial and different properties. In our assay system, this compound was able to inhibit the main proteases with the 6 micromolar, around 7 micromolar concentrations. So we predicted that it might be covalently modify the cysteine residues by using the by the site of the Michael acceptor site, but we were not sure that how exactly it is interacting, because we did not have the crystal structure for this one. And we have this catechol structure also is very important for making the hydrogen bond interactions. But its derivative, that is called salvinoleic acid A, where you have the additional unit of other catechol unit, though with the different lengths, was found to be less active when compared to its parent molecules. And we proposed that this moiety, which is highlighted in red color, could sterically hindrance in order to interact with the main protease. So that could be the reason why this compound is less active than its parent molecules. But though it is 28, around 29 micromolar activity. So more interestingly, we also selected some of this very active compound, and we tested their antiviral activity in colloid cells. The colloid cells is derived from lungs, so which is very natural to test the antiviral activity of the compounds, which also expresses the main cofactor, that is TMPRRS2. So before doing the antiviral activity, we tested the cytotoxicity of our compounds, because sometimes the cytotoxicity can be misinterpreted as antiviral activity, because ultimately you see the cell death. So first we wanted to make sure that they were not cytotoxic and the cells are alive in the tested concentrations. And then we tested the antiviral activity of our selected compound. You could see that here they are highly potent with the EC50 value of even to 1.3 nanomolar, which is highly effective. So we found that the robinetine and skutatelerine here, these two compounds were found to be highly effective against the SARS coronavirus 2 replication in colloid cells. So we also found that we really got very highly active antiviral activity. In the next slide, I would like to show about the other proteases, that is called papain-like proteases. So basically the many natural products have been investigated mainly with plant extract. They are responsible. It is responsible for cleaving 3 proteins. So it's mainly working. It's mainly working by disrupting the host antiviral response by removing the small proteins, that is ubiquitin and ubiquitin-like protein from the host proteins. So it blocking basically the interferon release by removing the ISG 15, so that the antiviral response will be diminished by the PL proteases. So many people also have been investigated on these viruses. I could show some of these natural products and or maybe the inspired compound, for example, the aldehyde and ethanol and different substituted esters. In the preliminary assay, they were also showed a good inhibitory activity, but I didn't see any further antiviral activity of this compound, and many more genomic amides isolated from the different plants also have been investigated, and many polyphenolic compounds, including isobava chalcones and different compounds have been investigated against the PL proteases, but I did not see many advanced studies that were active also in the cellular model. And these are some compounds. They did not show the exact molecular target, but they were highly effective in the cellular models, which is also very important, that they are inhibiting the virus replications. For example, the carolactones and the homoherringtonin and hematins, they were isolated from the different organisms and plants. And you see that the IC50 value is really highly effective, but they have a minimal cytotoxicity. But this could be the initial a point in order to develop more effective compounds without cytotoxicity. So I'm in the conclusion part. Natural products can be introduced as preventive and therapeutic agents in the different stages of the virus replications. For example, in the virus entry and maybe the uncoating process and the replication process, and you have the production of the inhibiting the RNA dependent and on RNA polymerases, and even the release of virus. And we have several examples as here that we have discussed. So we have also suggestion that to expand and to promote the research project on effective natural products in order to prevent the COVID-19, but can also be used for the future emerging virus infections. So it is not important that simply investigating the natural products against the virus, it should be also use of other natural products as effective coronaviruses. It is also important to identify the different other targets to combat the other coronaviruses. So again, the simple enzymatic test could not be enough to develop the drugs. So you need to really worry about to finalize the other properties, such as the pharmacokinetics and the pharmacodynamics properties of the molecules. And it's very important also to concern about the toxicity, though they are natural products, but you still see the toxicity in the cells. And based on the lead compound, it is very important to modify the original natural product compound, natural product compound, because sometimes the natural product has the complicated structure. So it is important it will be difficult to modify. So that it has to also be concerned, worried about to study the structural relationship in order to improve the activity of the molecules, and for the in vivo and clinical structure research development should be considered in order to develop as a promising drug against coronavirus infections. Again, I would like to insist the future outbreak infectious viruses. So finally, I would like to thank you, and thank you so much for your kind attentions. I would be very happy to answer if there is any questions. Thanks so much.
Yes, sir. We have some questions from our participants. Can
Yeah.
I dictate?
Please.
Yes, sir. Are any phytopharmaceutical-based formulations available today for treating the viral infections?
No, it is not officially available, but traditionally available. We see that the Chinese traditional medicine, herbal medicines, they have a different formulations. So which is possible, but it is not officially. I don't know if people are still taking this one. In the initial stage, of course it is, yeah, because many formulations have been suggested, but now we have the drugs and vaccines.
Okay, thank you, sir. Our next question is development of resistance and emergence of newer strains. How do we address this?
Development of resistance. I know it's basically the the original version which was emerged in 2019 is no longer available now. So what we are experiencing now is completely new strains. Okay, that can be only seen by the addressing the genomic sequence, whether it is, for example, we had a very big outbreak in India for the Delta coronaviruses. We didn't know that what was the reason for this sudden. Everybody thought that the coronavirus pandemic was slowed down and it will no longer be dangerous. But suddenly there was a emergency. There were the very high peak of infections. Then we didn't know what was the reason for this high, the immediate surgeons. Then they took the substance from the infected people, they compared the genome sequence, and then they compared to the original virus. Then they found that it is not the original virus anymore, but it is different, and it got the mutations in the spike protein, which really facilitate the viral entry. So this is how we could identify the strain, the strain. When you identify the strain, that means you have the chance of resistance because it is not original. For that, you develop vaccine. For example, if you developed the vaccine for the original strain, which is same, and when there is a mutation that it no longer be effective against your vaccine because the binding region will be modified, then the vaccine will no longer be binding to your spike protein, so it will not be effective. So this is how you get the resistance.
Yes, thank you, sir. Our next question is, as the virus protein mutated, can we design a molecule with the existing crystal structure of viral proteins?
Yes. For example, I was discussing about the main protease and pap-and-like proteases. They are viral targets. The problem with spike protein, the mutation, the rate of mutation with the spike protein is really rapid that you cannot really imagine. But if you target the virus proteases, something like PL proteases and main proteases, the chance of mutation with these proteases is very less. So you can develop drugs without much worry because the proteins will not be mutated. Otherwise, this will not be functionalized anymore. It cannot be functionalized for the cleaving the proteins. But of course, we can already predict the possible mutation of the amino acids even with these proteases, and based on that is also possible to address this issue. For example, the Paxlovid, which is approved for the SARS coronavirus 2 main proteases, which has the less chance of the mutations, but still the people have predicted the possible mutation of the amino acids in the active site, and they addressed, and we already think about what if there is any mutations in the active sites. So based on that, the molecule also can be utilized. So this is why the crystal structure also is very important. So basically, it is possible.
Yes, sir. Thank you, sir.
Yeah.
So now I would like to conclude the session.
Hello. Can I ask one question, sir?
Yeah, please.
Sir, thanks a lot first for your lucid presentation.
Yeah, thank you.
Yeah, during your presentation, you have shared some of the phytoconstituents activity, be it flavonoids, imodine, some polyphenolic compound.
Yeah.
Sir, whether these phytoconstituents are studied in vivo also, or only in vitro studies are being done?
I think so far only in vitro studies. the natural compounds are really good because they already have very low toxicity, but the problem is they have the ubiquitous activities that it inhibits many other proteins and enzymes as well. So probably you may lack the selectivity.
Okay, so current only from the basis on the in vitro and molecular docking studies, they are being treated as that having antiviral properties.
Yes, and some of them also they went to a cellular model, as I shown here, that they show the very good antiviral activity.
Okay.
But maybe if you are going to the animal model or something like this, then you will have a serious problem because they are not selective.
Yes.
Yeah.
Okay. Thanks a lot, sir. Thanks a lot.
Thank you.
Yes, thank you, sir. We have one more question from our participants.
Yes.
Could you please suggest if there are any clinical studies ongoing on using a natural product, sir?
So not yet. I think not yet. You know, in the at least not from the natural product of from the plant. Yeah. As far as I know, there is no natural product in the clinical studies, as far as I know.
Yes. Okay. Thank you, sir.
Yeah.
Now, I would like to conclude the session. Sir has explained wonderfully the life cycle of coronavirus. Then sir has explained about different approved drugs for the COVID, also about the natural products against coronavirus too. Sir has also explained about the virus entry into the human cell, then main protease as a target. What is a main protease? Their binding and their binding. Also, their cell lines used for SM inhibitors in a coronavirus too. Then sir has explained main protease inhibitors. Thank you, sir, for such a wonderful session. Kindly accept this certificate as a token of appreciation, sir.
So thank you very much. And again, I would like to thank you for the invitation. So I hope you get some information, though the interest of SARS coronavirus 2 is reduced.
Yes,
but
sir. It was really a wonderful session and we get very much knowledge from your session, sir.
Thank you so much. Yeah.
Yes, sir. Sir, please accept our token of appreciation, sir.
Yeah, thank you very much. What to do now with this? Should I download or?
No, sir, we'll share email.
Okay, cool. Nice. Yeah, thank you very much for this nice certificate. Yes, it's really honored.
Thank you, Dr. Pillaiyar. It was a great session.
Thank you very much.
More detailings into the replication cycle and all the products and their mechanisms. So excellent session, sir. Great.
Thank you. Yeah. So again, I would like to thank you and have a nice day. So thank you then. Bye-bye. If there is no more questions.
All the participants to start their videos, so we can take a group photo. I'm sure.
So it's done, right?
Just a minute, sir. Yes. Thank you very much.
Okay, then thank you very much and
Thank you.
have a nice day. Yeah,
Yes,
bye-bye.
Yes, bye.
Bye, sir. Thank you.
Sir.